Hello, my name is Brenham Wilson, and this is the suppression of mite reproduction as the ultimate defense mechanism against Varroa destructor. So Apis mellifera is the species of topic, or more known as the Western or European honeybee. Its name Apis means bee, and mellifera means honey-bearing. They are native to Africa, Asia, and Europe, but they can now be found on all continents except Antarctica. This is because humans introduced them to the new continents in the early 1600s. They are very important because they are the most frequent species of pollinators for crops worldwide. The next species of topic is the Varroa destructor, or more commonly known as the Varroa mite. They are tiny parasitic arthropods that live exclusively inside of Apis mellifera colonies. They feed off the honeybee larva and affect their development process, while they also can transfer bee viruses such as deformed wing virus or Kashmir bee virus. If not handled or reduced, there is a 90 to 95% chance that a colony will die if it becomes infested with mites. There is a trait called suppression of mite reproduction, or better known as SMR, that basically means that bees with the trait are resistant to parasitic mites. They are capable of detecting and removing mite-infested pupae from the cells, and they are also capable of detecting or even affecting the reproductive success in the mites. Which leads to the question, how does the SMR trait in Apis mellifera help colonies protect themselves against Varroa destructor? So here was a study done that showed the SMR traits in queen bees. On the graph on the left, you can see that there's 3 different colonies, each colony with a different queen. One with a full-bred SMR queen, one with an SMR free-mated queen, or a 50% SMR queen, and a control queen with 0% of SMR traits in her. Each colony was infested with varroa mites and inspected after 115 days. As you can tell, the control group had a high mite population after the 115 days, which makes sense because they have no known defense against them. On the other hand, the full-bred SMR queen in her colony had very little mite population. When you look at the second group with the SMR free-mated queen, there is a substantial amount of mites left, but it's nearly 50% less than that of the control group, or the control queen in her colony. When you look at the table on the right, it compares the free-mated SMR queen to the control queen. And when you look at the final mite population of the free-mated SMR queen of 1,059 in her colony compared to the control queen of 2,711. Also, the percentage of non-reproductive mites in the free-mated SMR queen in her colony was 51% to 29% in the control queen's colony. Overall, you can say that colonies with at least 50% SMR trait are significantly more resistant to varroa mites than those with no SMR traits at all. Next, here is a study that shows the behavioral trait of recapping and how it is associated with SMR. So, recapping is a behavioral trait of honeybees where cells with developing pupae are uncapped, inspected, and then recapped without removing the pupae. So data was gathered from 3 different SMR colonies from 3 different countries, France, Norway, and Sweden, and then 1 control group with no SMR traits at all. The study measured the proportion of recapped cells in each colony and the number of viable female mites that were observed afterwards. When you look at the scatter plot, overall there is a downward slope, which supports the statement that colonies that show more frequent signs of recapping behaviors have lower mite populations than colonies that don't. The final study I looked at tested the reproductive success of varroa mites without adult bee interference. Infested cells from an SMR colony and a colony bred for hygienic behaviors were placed in incubators at 34 degrees Celsius. After 10 days, the reproductive success was recorded for each cell. The mites were significantly less fertile, and the number of viable female offspring for the mites was also less. In conclusion, Colonies with as little as 50% of the SMR trait can provide a significant level of resistance to Varroa mites. The recapping behavior, which is associated with SMR, also promotes defensive behaviors against Varroa destructor. Overall, Apis mellifera with the SMR trait have an effect on the reproductive success of Varroa destructor. Breeding for the SMR trait provides a safer method for defense against Varroa destructor compared to other methods such as chemicals, which can be harmful to the bees. It also gives a long-term solution for protection against Varroa destructor.